What specific problem does your method seek to address in understanding the training process of language models?
Our method seeks to address the problem of understanding how the intricate computational structures of language models develop during training, akin to the embryological development of organisms. By employing UMAP to visualize the low-dimensional projections of susceptibility vectors associated with token sequences, we aim to reveal the internal structure of the model and how it evolves over time. Specifically, we focus on identifying and interpreting patterns in token susceptibilities, which reflect how different components of the model interact to predict the next token. This approach allows us to visualize significant structural developments, such as the emergence of the induction circuit and the formation of a spacing fin, providing insights into the model's computational capabilities. Ultimately, our work contributes to the broader goal of structural inference, helping to elucidate the complex dynamics of language model training.
How does the proposed method of visualizing susceptibility vectors work? Illustrate the steps taken to obtain these visualizations and explain the significance of UMAP in this context.
The proposed method of visualizing susceptibility vectors involves several key steps. First, for each token sequence XY, a susceptibility vector, eta_WXY, is computed, which captures the per-token susceptibilities across the model's attention heads. Next, these vectors are collected from a dataset of token sequences sampled from the training distribution, forming a point cloud in math_BB_R_H. To visualize this high-dimensional data, UMAP, Uniform Manifold Approximation and Projection, is applied, which reduces the dimensionality while preserving local topological properties, allowing for a clearer representation of the data manifold. The significance of UMAP in this context lies in its ability to reveal the internal structure of the language model as it develops over training, highlighting patterns such as the stratification of token types and the emergence of computational structures like the induction circuit and spacing fin, as illustrated in the colorful rainbow serpent visualization. This approach not only aids in understanding the model's behavior, but also provides insights into how different components interact during training.
Can you provide a simple example demonstrating how a susceptibility vector is computed for a token sequence and how this relates to the model's predictions?
To compute a susceptibility vector for a token sequence, we consider a specific context, X, and a next token, Y. For instance, if X is the cat and Y is sat, we associate a susceptibility vector, eta_WXY equals chi_C_1_XY, L dots chi_C_H_XY, where each entry, chi_C_J_XY, measures how the prediction of Y given X is affected by perturbations in the weights of the model's components, C_J. This vector captures the covariance between the model's prediction for Y and the behavior of the components in response to changes in the weights, indicating whether those components are expressing or suppressing the continuation of X by Y. For example, if chi_C_J_XY is positive, it suggests that component C_J is involved in suppressing the prediction of Y, while a negative value indicates that it is expressing the prediction. Thus, the susceptibility vector provides insight into how different parts of the model contribute to the prediction of the next token based on the context.
What theoretical or practical advantages does using UMAP for visualizing susceptibility vectors offer over traditional methods of examining language model behavior?
Using UMAP for visualizing susceptibility vectors offers significant theoretical and practical advantages over traditional methods. Firstly, UMAP provides a low-dimensional representation that accurately captures local topological properties of the data, allowing for a clearer visualization of how susceptibility patterns develop over training, as evidenced by the stratification of token patterns in the UMAP projections. This is particularly useful for observing the emergence of complex structures, such as the induction circuit and spacing tokens, which may not be as easily discernible through conventional analysis methods. Additionally, UMAP's speed facilitates rapid analysis of large datasets, making it feasible to track changes across multiple training checkpoints, as demonstrated in the paper. Furthermore, the visualizations produced by UMAP, such as the rainbow serpent, offer an intuitive and interpretable representation of the model's internal structure, enhancing our understanding of the interactions between different components of the language model. Overall, UMAP's ability to reveal intricate relationships within high-dimensional data makes it a powerful tool for structural inference in neural networks.
Describe how you validated or tested the effectiveness of your method. What datasets and metrics did you use, and how did they support your findings?
In this study, we validated our method by applying UMAP to the susceptibility vectors derived from a 3M parameter, attention-only transformer model, trained on a subset of the piled dataset. We focused on the per-pattern susceptibilities and their evolution over training, particularly observing the emergence of structures like the induction circuit and the spacing fin. The effectiveness of our method was supported by visualizations that showed clear stratification of token patterns in the UMAP projections, which were quantitatively corroborated by tracking changes in per-pattern susceptibilities at various training stages. Additionally, we compared results across 4 different seeds of the model, demonstrating a high degree of universality in the observed structures, which further validated the robustness of our findings. The consistent patterns observed in the UMAP visualizations and the corresponding susceptibility metrics provided strong evidence for the internal computational structures identified in the language model.
What limitations or challenges did you encounter in your study, particularly regarding the scale of the model or the tokenization strategies employed?
In our study, we encountered several limitations primarily related to the scale of our model, which is a 3M parameter, 2-layer attention-only transformer. This raises the question of whether the UMAP of susceptibility vectors can effectively reveal structure in larger language models, as our findings may not generalize beyond this specific architecture. Additionally, our interpretability method relies on UMAP for visualization, which can distort global distances. Thus, the geometry of the rainbow serpent should be interpreted with caution. Furthermore, the prominence of the spacing fin we identified is likely influenced by our specific tokenization strategy, as different tokenization approaches could lead to varying learned structures. These factors highlight the need for further investigation across different model scales and tokenization methods to validate our findings.